Hello, I'm going to show you a collection of objects that are related to gene expression, and these are actually Xenopus laevis at various different stages. That's a type of frog that's used in experiments. So you can see here, this is at an early stage. This is stage 26. This is stage 39. And down here you can see there's metadata associated with each one of these images, and that means we can do interesting things with these collections. So for example, we might want to be interested in looking at different stages of their growth. So here we can do a histogram, and we can narrow it down to stage 39 in this case. If we wanted to do that slightly differently, we could look at this in the graph view, and look at these as different histograms, and we can drill down to any of these particular stages. So we're going to look at stage 39 in this case, and then we're going to look at how a concentration of a particular gene can be altered using a blocking agent called a morpholino. And we have these concentrations logged, so these experiments have been done at various concentrations on various different genes to understand how much concentration of morpholino we need to be able to block those genes. So let's look at, we're on stage 39, let's have a look at that from the point of view of the target gene. So we can see here we've got several genes that we're looking at in this particular one, and we can look at the AMI gene, and then if we then look at this in terms of the concentration of morpholino, that's the blocking agent. So you can see here this C is the control, and you can see there's a fairly wide range of expression here, but this is the control, so this means that the morpholino isn't doing anything. And you can see here in these particular concentrations that again the morpholino doesn't have much effect, but at these concentrations that seems to work pretty well. So this is a good example of the subtlety between imaging that even using machine learning would be quite difficult to pick out. So let's just reset all the filters of that. So just to show you a different way we can look at data in Zegami. We can go, for example, from a type of plot called a TS&E plot. So let's go back to grid and do the filter. So we've done pre-calculated TS&E. So it's basically this is unsupervised machine learning on all this data, and we can compare it, say, to the first component of the TS&E versus the second component. And what this allows us to do is then this groups together really nicely different classes of images. This is without doing any particular sort of like training. You can see these are all kind of like coming up. And in Zegami, one of the nice things we can do is actually try this between different facets. So for example, if we click on stage here, this is showing the different stages of growth here. So if we look at this, because it looks like an interesting outlier, you can see here somebody's left the tweezers in, and this is in the stage 26 of the experiment. So one thing we could do then, we can tag that and say that's a bad one. So we can do that and say that's a bad one. and we can do much more fluid tagging as well. Say for example we remove this filter, we can do tagging on different individual groups and sort and filter on those. So for example, I could do view types, graph, maybe drill down, and maybe I'm kind of interested in some of these. Maybe I think there's obviously some weird photos here which could be detrimental to any kind of classification you were doing. So we could also label these were bad as well. And these can then be exported as a table to a machine learning. So you could actually get the machine learning with supervised learning to be able to see these. So we should see this is stuck on stage 39. Okay, so you can see all these different tags here. Okay, that's the end of this particular demo.